lecture test, you just need to know the four most common ones. Okay, so just to make it a little easier, you just need to know the four most common ones. And to kind of show you that, we're going to show you 4 control organisms growing in thioglycolate media. Thioglycolate is a reducing media that gets rid of the oxygen in the broth. So you might remember there was a little red layer at the top of these thioglycolate tubes. You guys remember that little red layer? Remember I said don't shake these. That goes for now too. It's very important that when you go in there, you don't like slam stuff around because you'll disrupt the growth pattern. So look up here for a moment, guys. If an organism is like E. coli, if you remember from the lectures, E. coli was my example, facultative. So what does facultative mean? It's flexible. It'll grow with oxygen and without. So you're going to see E. coli growing everywhere in here. If an organism, on the other hand, is an aerobe, where is it going to grow? Aerobe. At the top, only at the top. An aerobe grows only at the top. What about a microaerophil? Micro, little bit of air. A little bit below the surface. It'll kind of hang out right at the bottom of where there was that red layer. Keep in mind that some organisms utilize the oxygen in that layer, and you won't see that pink anymore, okay? So that means you remember where you first saw the pink at the top? It may not be there anymore because the organism uses the oxygen up, okay? So if it's a microaerophil, it'll sit down below the surface. You'll see a little layer of growth. What about if it's an anaerobe, against oxygen? Where's it going to grow? Yeah, but keep in mind, guys, it's not just going to grow in this little pellet down here. Because remember, about 2/3 of the tube does not have oxygen. So it will be able to grow anywhere in there. It's not just that it's not going to sit like at the bottom. Just keep that in mind. It'll grow almost everywhere except for at the very top. So if you take your tubes out and you bounce them around and knock them around, you're not going to see that. You're not going to see the pattern. So you literally want to be kind of careful. And I'll bring them out here and try not to trip when I'm carrying the tray out. I'll try to be careful. But again, just don't shake those. So you want to look and see where you see growth and indicate that growth. I suggest, guys, don't use a yellow or white colored pencil. Don't use that. Use something darker so you can actually see where the growth is. Okay, you see what I'm saying? Sometimes students will draw, it's a white piece of paper and you're using yellow or white and you can't really see that. So do something that you can actually see where the growth is in your drawings, okay? So you remember the patterns. So you're drawing your gram positive and your gram negative, and you're hopefully noting where the growth is. Is it at the top? Is it everywhere? Is it just below the surface? I will not interpret your unknowns for you. So don't even ask, or just go. So you need to look at each other's and look at the controls to compare them. I won't interpret it for you because it's your unknown project. You have to observe. Don't worry, it's not going to mean the end of your grade if you misinterpret it, but do your best to interpret it properly. Then we're also going to bring out a candle and a brewer jar that you can compare the growth of the organisms that you saw in the thioglycolate. So let's see if we remember this. A candle jar is made to allow the growth of what kind of organisms? Microaerophils. It actually, literally, you'll see, has a candle in it that burns most of the oxygen but leaves a little bit of oxygen in there. So the candle jar is designed to enhance the growth of microaerophils. However, guys, what other organisms might grow in a microaerophil? I mean, in that condition in the candle jar. There's a little bit of oxygen. The aerobe will grow too. It's not going to love it, but it will still grow because there's still a little bit of oxygen. So the candle jar has a little bit of oxygen. So the microaerophil is going to love it. The aerobe is going pretty okay. And then finally, what about the facultative, guys? Sure, it's flexible. So what won't grow in the candle jar? The anaerobe. The anaerobe should not grow in the candle jar. The anaerobe won't grow because there is a little bit of oxygen in there. However, there's a brewer's jar, and the brewer's jar is so named because it's what brewers do. When brewers make beer, they want anaerobic conditions so fermentation will take place. That's why they're called brewer's jars. So you're not making beer here today, sorry, that's another class, but the brewer's jar has no oxygen, zero. So what type of organism is it designed to allow the growth of? Anaerobes, against oxygen. They're gonna love the brewer jar because there's no oxygen in there. But what also will grow in the brewer jar? Facultatives, okay? So facultatives are gonna grow in both of these jars. Anaerobes will grow in the brewer jar, but not the candle jar. Aerobes and microaerophils are going to grow in the candle jar, but not in the brewer's jar. So you have to kind of sort all this out in your head. So what you'll notice is we'll bring these out. They're the jars, you can't open them, and they have tubes in them. You have to kind of hold them up to the light and look at them, because the growth of a couple of these organisms, let's see, this one, and this one are rather transparent. Okay, that means they're not real opaque, so you have to kind of hold it up to the light to see the growth. Okay, the bottom line is you're going to indicate right here, guys. Woo, it says summarize the results of the table, the growth of the organisms in the candle and brewer jar using the following scale. So in the table, you're not going to draw the tubes. You're just going to put no growth, some growth, ooh, lots of growth. Okay, so see this little thing here, that's what you're going to use for that. Okay, and then finally the other lab that we did, that we set up, was motility. Anybody have that page? I'm depending on you guys, somebody have the schedule in front of me. 85, thank you so much. So guys, remember for motility, you were to set up your gram-negative unknown, and you did it with a stab. Do you remember that? You took a needle and you stabbed it into the media. And today you're going to compare your gram-negative unknown with two controls. Okay? So if the bacteria is motile, they have flagella. If they have flagella and are motile, they'll swim away from the stab. So you'll see that the whole media will have the growth. And the way that you see growth, you'll see is there's a red color. There's a dye that's put in the media that you can see. You'll be able to see by comparing your unknown to the two controls. So the control that's positive is Proteus. So just FYI, Proteus, the name Proteus is after the god of the sea. Okay, so like in mythology, there's a god called Proteus, okay? And it's a god of the sea. So Proteus is the bacterium of swimming. It's the most notable swimmer, notorious, I should say. It will, if you have it as an unknown, it'll swim all over your plate and make it really hard to isolate it. So it's actually in a clinical setting, can cause, for example, diarrhea. No, it doesn't cause diarrhea. Proteus causes urinary tract infections. And you know how it does it? It swims up your urethra. So, you know, women are encouraged to, when they use the restroom, they're supposed to wipe front to back. That's so that you don't get any fecal stuff near your opening of your urethra. Because Proteus can live as normal flora in your intestines. And if it gets to the base of your urethra, it's off to the races, up towards your bladder. And if it doesn't get stopped in your bladder, it's gonna keep on going and swim up to your kidneys. So that's why sometimes these motile bacteria can be a problem in the clinical setting, because they can swim, not just in a petri dish, but in your body, okay? So men don't usually, when they're young, don't usually have a problem with urinary tract infections, okay? And that's just because the plumbing is a little different, and fecal matter doesn't as easily get into the opening of their urethra. So usually men don't start getting urinary tract infections until they're old, and they have an enlarged prostate gland, and then they can't completely empty their bladder, blah, blah, blah. So none of you guys, don't worry about that. You don't have to worry about that for a while. So bottom line is you're gonna be drawing your gram negative, looking at the two controls. I'm telling you already, Proteus is our classic positive control. You'll see the red growth is throughout the whole tube, whereas the staph gets sort of stuck where you stab them. I won't interpret your unknowns, okay? So students always want, oh, is this a positive or a negative? I'm like, mum's a word, look at the controls, okay? So don't think I'm being mean, I'm just trying to get you to trust yourself a little bit. I am a little mean, but I want you to trust yourself and then feel like, you know, you can do this, okay? All right, so I'm gonna get those out. You're gonna be sketching them in your notebooks, and I'm gonna try